Introduce the next speaker, Dr. Saeed Aoufouchi. Here he is, yes. He's the head of the Genome Plasticity and B-Cell team at the Gustave Roussy Cancer Center in France. And he will talk to us about mutagenesis in adaptive immunity, unique setting where error becomes a quality.
Okay, thank you very much. I start just to thank the organizer for giving me this opportunity to share with you some of our recent data, and also I have to really to give special thanks to Dr. Gada, because she did want to bring me from my adoptive continent to my mother continent to see what's happening in science in Africa, and thank you, Gada, because I think it's a great pleasure. So I just start with acknowledging that the people who did the work, because I feel that we'll run out of the time on have this, and I think there is an important slide. So that is the people in my group is doing this this work. And also I will thank the institution and the funding, because the funding are important for all this work to be done. And finally, as you know, science, to do science, you need to have some nice interaction and collaboration with people, and I thanks all my friends and colleagues for that. So we heard for the last two days about the human genetics, human disorders, and I want just to share with you some thinking about getting back, take you back some steps and think what is the origin of all these disorders. And I think all of you will agree that the somatic mutation is one of the important things of these disorders. So the somatic mutation will concern all the cell of the body, and they occur throughout the life, so we can't stop them. That is part of our life. And the effect of the mutation will vary a lot, from no effect to mild effect to very serious effect, which the more serious ones are compatible with life. And we all agree that they are the origin of all the human disorders. And of course now with all this collected data from the sequencing data, we all agree that all the cancers are caused by somatic mutation. And that is just one example from one Nature paper, which we have been collecting, sequencing many cancer cells, and all the cancer have a load of few to very heavily mutated cancers. So our understanding for the biological process generating those mutations is very limited. So we know very little about what is causing this mutation. And that is quite remarkable, because you have to think that if the cancer is caused by mutation, so understanding the causes of the mutation is understanding the causes of cancer. And I just to highlight what we know about the origin of some mutation. I think that we know that the cell, the environment of the cell is, and especially the environment of the DNA is quite toxic. The DNA is exposed to highly toxic environment, make thousands of damage on the DNA. If it's just not repaired, you will get mutation. The DNA replication is very risky timing, because we duplicate a billion of nucleotides, and we can make a mistake, and if the mistake is not corrected, we'll acquire, and I think we estimate between 1 to 2 mutations by cell cycle. And if we get exposed to radiation, again, the DNA is get damaged, and if it's not repaired, it will occur mutation. If we get exposed to the chemical environment or to the toxic smoking, we will ultimately damage the DNA and we can get the lung cancer. Alpha toxin is issue in Africa, is something which is concerned part of Africa. It's chemical which is produced by fungi. It gets contaminated in maize. If somebody will eat the contaminated maize, ultimately will go to the liver and can cause the DNA damage, and ultimately can cause hepatocellular damage. As we know, all this the DNA, all these cells are highly exposed to the continuous DNA damage. We have this many mechanisms of surveillance. We are dealing and repairing every day and every time all those damage. And among the DNA repair mechanism, the sensors and checkpoint and tumor suppressor gene, I think all you are aware of them, and also the immunity checking every day to deal with those toxic events. And paradoxically, if you have some damage in one of those components of those different pathways, the cell is become more prone to mutation, and you accumulate more mutation, and you get to the cancer. So those mechanisms also have to keep checking on them. So there's only happy faces on mutation, because there's sometimes some people are lucky they can acquire some good mutation which can help them to deal with some disease. But what I'm trying to show you today, which is the specific of my group, is to talk about somatic hypermutation of immunoglobulin gene. So I'd like to have a little bit more attention in this part, because normally I lose half of the people in the two coming slides. So the somatic hypermutation will concern the immunoglobulin gene. Is the gene which affect the code for the antibodies, just concentrating the B cell immunoglobulin. So the gene are assembled from VDG segment through what we call the VDG recombination, VDG for heavy chain and VJ for light chain, and that is done by two specific hematopoietic gene, RAG1 and RAG2, together with the non-homologous androgenin molecule, and that which will give a B cell to express a functional immunoglobulin at the surface. These parts happen in the bone marrow, and it's not dependent of the antigen. So now that when the cell become mature, it will leave the bone marrow to the periphery, and when he encounters an antigen, he will start another diversification process, and that is what we call somatic hypermutation and class-switch recombination. These processes are initiated by AAD, with activation and ubiquitin deaminase. Again, this protein is the only protein which is specific for the immune system. All the rest are ubiquitous protein. The somatic hypermutation is mutation which target the VDG gene, especially it's very specific and targetable mutation. The somatic hypermutation is the underlying what we call the affinity maturation of the immune response, of the humoral response. This is happening in germinal center, which is some microenvironment where the B and T cell will collaborate to have a better response. So the B cell, the activated B cell, which express naive antibody on the surface, will go to this germinal center, start proliferating, and doing the somatic hypermutation, and then we go through selection. The cell which made the best mutation will be selected and end up at giving the best cell, plasma cell, differentiating plasma cell, which will produce antibody able to clear the antigen. But also a part of it will be differentiated into memory B cell, which can be activated when you're challenged by the same antigen for the next time. And that is the principle of vaccination. So if I put it just as a cartoon, the B cell, it's naive B cell. So the antibody, you have to imagine the antibody created in the bone marrow does not recognize the antigen, but they have some affinity to this antigen. You get the help, this B cell will get the help once you recognize this antibody from a T cell and start to mutate the antibody and be selected for the best one. And you end up with a B cell which express a high affinity antibody, specific antibody. Have to keep in mind that it's the somatic hypermutation. It's happening. It's very mutagenic process targeted to the immunoglobulin gene, and he's about 1 million times superior to the background mutation. So it's very high mutational process. So how it's happened? That I will just go through quickly about what we discovered we and the other about the mechanism happening. So you start the mechanism by activating and use cytidine deaminase, which deaminase the cytidine in the immunoglobulin gene. It's become uracil, and this UG now can be taken by two mechanisms, either as uracil, and that is the normal basic fusion repair, which can generate a mutation at GC bases. And this UG can be taken also as a mismatch, and we will deal it by the mismatch repair protein, which will incorporate a very mutagenic polymerase, which will do the mutation in AT bases. So we'll summarize this this in one slide, which we spent the last 15 years to address this question about the mechanism, how the cell can tolerate that much number of mutations, and how can deal with this number of mutations. So, as I said, the AAD will initiate this system, but Pol Eta will add the mutation as with the help of the mismatch repair protein, and make the AT to GC mutation. So when we delete, so we make a mouse model, like in either Pol Eta, MSH2, or both of them, and you will see when you remove Pol Eta, with the AT mutation will drop. Again, with the mismatch repair, this one will drop again. And when we remove all of them, we lose practically all the AT mutations. So the conclusion from this work is the Pol Eta, together with the mismatch repair and somatic hypermutation, are responsible for 100% of AT mutation. So we know that it's, the system can give us a very good clue how happening those mutations. Not only this, we observe this one in the mice, because we have in the a situation in human which is XPV, and that is the XPV, it's variant, the Xeroderma pigmentosa variant, which lacking the Pol Eta, and again we find the same AT mutation defect on those people. So we know this Pol Eta is important for this. So I think the work in the immune system show another role of polyeta. We know this polyeta, as you know, it's important for the error-free repair across UV lesion. But in the immune system, we don't have any lesion, and one mismatch repair coopted polyeta will introduce AT mutation. And now we describe for the first time a signature of the polyeta and mismatch the somatic hypermutation related polyeta signature. And thus, now, if the all work which we've been done is to show that it's this process, which is physiological process in the immune response, can be used as the signature to see if they have some, if we can target some other gene other than the immunoglobulin gene, if that can have some deleterious effects, or we can have some malignancy. And we checked for this. We can find this signature in the B-cell lymphoma, and we did, yes, a few years ago, we find this mutation in the diffuse large B-cell lymphoma, and find this mutation in many oncogenes, PIMPAX, RHO, and c-MYC. And those recently some work from two groups. We show this mutation. We can find it in B-cell lymphoma and in CLL. And a recent work, which has come just a few months ago, show clearly the signature of somatic hypermutation, polyeta-derived mutation, are responsible for more mutation in the lymphoma, and some of them in the melanoma. So what I want to take home message from this one is we learned from the somatic hypermutation in immunoglobulin gene a signature, a very strong signature, which can be at the origin of many malignancy in the human the B cell. And well, we have maybe some other things to learn from the immune system in terms of this driver mutation. And I think the other thing which to think about it is the cause of somatic mutation should be amongst the priority of this for you scientists and the geneticists, maybe because in long run it will be used for the cancer therapy or maybe just for prevention. And thank you very much.
Thank you very much, Dr. Saeed, for showing us how the error becomes a quality sometimes. And thank you for keeping the time, which gives us the chance to have a couple of questions. Please.
Saeed, I think you'll have trouble hearing me because of the background noise there, so maybe you move this way. So I just want to ask a question. Thanks very much for that wonderful presentation. What would you care to speculate whether a phenotype that has got a germline mutation in a mismatch repair gene, you got a phenotype which has a germline defect in a mismatch repair gene, either heterozygously or homozygously, what effect would that have on, for example, the phenotypes you talked about? Because some of these, so this would be something like familial colorectal cancer because of mismatch repair genes, and the rare instance where you get a homozygous mutation causing CMMRD syndrome, and they do have hematological disorders.
Great question. I think we know from the people working with the mismatch repair that is the absence of mismatch repair, as we said, is very highly mutagenic, and it's just most of the now mutation in the gene which comes with the mismatch repair, the polymerase is epsilon. It's very highly mutagenic. So we anticipate, because we have some work, the setting of the immunoglobulin gene is because you can see in very small gene and a huge number of mutations, and that you can't see it elsewhere, and that's the sitting is very good. So we look at for the mutation either in homozygote or heterozygote. The advantage of having this system, because all those mutations are driven by mismatch repair related. Either the mismatch repair have defects and you can have the mutation and you can anticipate the mutation mismatch repair, or you can have, because in the immunoglobulin gene, and that which we call, because that also we describe what we call the non-canonical mismatch repair, because in this case, because you see the mismatch repair will work in post-replicative repair. And in case of somatic hypermutation, you work in G1. So the B cell is doing something completely different. It's really coopting the mismatch repair, which normally work with the very high fidelity polymerase. In the B cell, we're working with polyeta, which is making hundreds of mutations. That is very mutagenic problem. But the B cell needs mutation to adapt to the antigen, and it got to just a few days to make good antibodies. And so now we have to look at the mismatch repair either as defective mismatch repair for some mutation, and you can see what the consequence. And the other view now is the mismatch repair will get coopted outside of his role, and that will recruit not the good polymerases, but the other polymerases, and that we will have another panel of mutations which we can anticipate and can create.
Thank you very much.